In this video, we're going to explore proper use of the microscope. Of course, you'll have a better idea once you get your hands on a real microscope, but for now, it's important to understand the theory behind it and how it works. So for safety, you have to make sure that you're always holding the microscope with two hands. You should always hold the microscope under at the base there, and then the other hand should be holding it on the arm. Whenever you're walking with a microscope, always walk slowly and carefully. Never run with it, of course. Never hold two microscopes at a time. They're not dumbbells, they are microscopes. You should always, when using the microscope, start with low power. Once you've started with low power, the idea is you locate your specimen under the low power by using your coarse adjustment knob. Once you've located the specimen, you're able to change the power to medium and high, and then once you're on medium and high, you can start using the fine focus, and you'll no longer use the coarse adjustment knob because you don't want to damage the microscope. If you ever need to clean a lens, it's important that you don't just use regular paper towel. You have to use lens paper so it doesn't scratch the lens. And there is a specific way to put away the microscope. Typically, when you put away the microscope, what you want to do first is put the low power objective lens back into place. You want to make sure that you bring the stage all the way down. So once your low power is in place, you use the coarse adjustment knob to bring the stage back all the way down. You want to remove the slide and put away your slide in the designated area. You don't want to leave the slide on the stage. That's important. And clean the slide if it's not a prepared one. If it's a prepared one, if it's not a prepared if it is a prepared slide means that it's one that we ordered for you to use. You would not clean that because it's a prepared slide. But if it's one that you made yourself, where you got a sample, you put a drop of stain on it, that you would have to clean. So if it's a prepared slide, you just return it to where it was, where your teacher tells you to put it. If it's one that you made on your own, you have to clean it, and there's a proper cleaning procedure we'll let you know about as well. It's also important that you turn off the lamp, unplug the microscope, and wrap the cord around the base. You do have to be careful when you're bringing a microscope back, because what can happen is sometimes as you're carrying it back with two hands, the bottom can be a little bit hot because the light was on. So just check to make sure it's not too hot as you're carrying it back. So those are the basics. When you use the microscope, we'll observe that you're doing these things. For example, are you holding it with two hands? When you're putting it away, did you follow these steps? When you're using your microscope, are you on low power and using the coarse adjustment knob? Are you using the fine adjustment knobs on the other powers? So those are all things you need to be very intentional about as we're watching you. So just into more detail here, how to carry your microscope. As we mentioned, always with two hands, one hand on the arm, one hand on the base. Walk slowly and carefully to your destination, and make sure that your microscope is on low power as you're about to use it. So this is the setup. Before you start, you make sure your microscope is on low power right here by turning the revolving nose piece. Bring the stage as low as possible before you start using it. So use the coarse adjustment knob to bring the stage down as much as possible. Unwrap the cord and then plug in the microscope, because you'll want to turn on the lamp to view your specimen. Once you have your slide, if it's a prepared slide, so one that's already made for you, you'll put it under your microscope, under the stage clips, aiming to get the specimen in the center of the stage where the light comes out of. Again, make sure that essentially it's in direct view of the objective lens, and you'll turn on the lamp. So this is what it looks like when your specimen is on the stage, ready to be viewed. It should be on low power, so this was later on, once they found it on low power, they changed it to a higher power, but you'd start off at low power. So now you're going to try to find your specimen. What you do is you put your eye on the eyepiece lens, and you turn the coarse adjustment knob to bring the stage up slowly until you actually see your specimen. So as you bring the stage up, eventually you will see the specimen come into focus. And what you can do is, once your specimen is in focus, if it's blurry, you could use the fine adjustment knob to make it a little bit less blurry. But the whole point is you did this at low power first. Once you find it at low power, then you're able to switch to other powers to find the cell and to zoom in a bit more to get more detail. So if you want to find the image at medium power, all you do is you spin rotate the revolving nose piece, or you spin it around to get your medium power into place. It has to lock in. Look into the eyepiece, and you'll see your specimen likely looks a little bit blurry now. That's okay, it's because you zoomed in a bit more. To make it less blurry, just use your fine adjustment knob. Don't use the coarse adjustment knob, because remember, the coarse adjustment knob would bring the stage up and you can smack it into the lens, which you don't want to do. So once you find that at medium power, you are now seeing a larger version of that image. So here's our question. Do you see more of your specimen or less of your specimen at medium power, and why? So think about this carefully. You actually see less of the entire specimen. You see less of it. And the reason you see less of it is because you zoomed in. So yes, you're seeing more detail, but you're seeing less of the entire organism. It's almost like if you're taking a picture of a few people from far away. You can see, like say you have a group of 10 people, you can see all 10 people in your camera if you're zoomed out. But when you zoom in more and more and more to just a few people, and you see less of all the people that are there, but you see more detail of the people that you zoomed into. Or it's like if you're taking a selfie. If you're taking a selfie from far away, you can see your entire body, or a bigger chunk of your body. But if you zoom in, let's say just your nose, then you see more detail on your nose. Maybe you see a zit there, but you see less of you as the organism. So the more you zoom in, the more parts of your organism might get cut off because you're zooming in more and more and more and more and more. So think about that as you're trying to zoom in to see your specimen under the microscope. If you want to view your specimen at high power, you would basically do the same process, except you would turn the revolving nose piece to the high power. You would once again have to turn the fine adjustment knob to make it less blurry, never the coarse adjustment knob. And similar reasons, you zoom in more and more and more, you see less of the entire organism because you zoomed into a more specific part of that organism. It becomes more detailed, but again, you see less of the entire organism. One more important thing to mention as well is that if you're trying to zoom in to see a cell, when you're looking into the microscope, it's a bit hard to see now, but when you get the time to play with the microscope, you're going to see that your cell, there's an arrow attached to the microscope. There's actually an arrow in the field of view. You can move the slide with your hand so that the arrow points at your cell that you're interested in. And that's the area you're going to zoom into. If your arrow is not pointing at the cell that you're interested in, well, then you're going to zoom into some other area and you'll lose the cell that you were interested in to begin with. So it's like if you're taking a picture, again, of a large group of people and you want to zoom into one specific person. Well, if your camera is not pointed the right way, you're going to zoom into the wrong person and you won't get a photo of the person you want to get a photo of. So that you'll see more when you get hands-on practice with that. Once you're done analyzing your specimen, usually you'll do a drawing of your specimen. You're going to start putting your microscope away. As we mentioned, you're going to return to the low power objective. You're going to remove the slide and return it to the appropriate area. You're going to turn the coarse adjustment knob to bring the stage all the way back down. You're going to turn the lamp off. You're going to unplug the microscope and you're going to wrap the cord around the base, or sometimes there's an extra part on this part here where you can wrap the cord around, but if that's not there, you'll wrap it around the base. Don't pull on the cord. Make sure you grab it from the base of the plug. Then you're going to bring the microscope back to the designated area, usually a cart or a station of some sort, with two hands, one hand on the arm and one hand on the base. One more thing to also mention. When you're observing the specimen, so let's say you're over here. Let's go back actually here, back to our slide we were at before. And you're working in groups, and you want a friend or your peer to see the specimen that you were looking at. Don't lift the microscope up and drag it and give it to your friend. What you can do is you'll notice that the part that's connected to the eyepiece, this part right here, that can actually swivel. So you can actually just revolve that, rotate it around, spin it around to move where the eyepiece lens is. So if the eyepiece lens is over here and you're looking in here, you can just rotate it and your friend can see it here, so you don't have to move the entire microscope. That can damage it. It can also make you lose where your image is focused, so you'll lose the cell you're looking at. Whereas here, you can just spin that around, and then your friend can see it without you actually ever lifting the microscope across a lab bench. So in this video, we briefly learned how to use a microscope, starting at low power, finding the specimen, then going up to medium power and high power, and using the fine focus knobs to focus the image. What we're going to do in the next video, we also learned how to put away the microscope properly and how to hold it properly. What we're going to do in the next video is take a look at the concept of magnification and do some calculations with that as well.